Okay, so I am the next presenter, and so we've already heard a little bit at this workshop about the microbiome and the role that it plays in this field, and there's clearly a lot of scientific as well as public interest in microbiome science, and I was drawn to this field of microbiome science as a postdoc coming out of a zebrafish developmental genetics background. And my lab over the last number of years has been trying to use the zebrafish as well as mouse and human models to understand the role that the microbiome plays in nutrition and immunity and other aspects. So in my remarks today, what I'm going to do is briefly summarize two stories, both published, but I think are the best examples for this group of how the microbiome affects host nutritional physiology and how dietary nutrients affect the microbiome. And then finally, at the end, I'm going to offer some challenges that are presented by microbiome research that I think are somewhat unique within this group. So the first study that I'll talk about is an effort that we made a few years back to try to understand the impact of dietary nutrients on zebrafish microbiome assembly. And we chose dietary fat for reasons that will be apparent later on in the talk. So the study design is shown here. We took a large cohort of zebrafish embryos, split them up into a number of different tanks. We actually included in this treatment some tanks that had no fish in them. This will come back later on. And then we sampled gut microbiomes as well as the water microbiome from these tanks at several different time points during development. So the diets that we used here to look at the impact of dietary fat were developed in collaboration with an R&D group at Ziegler. I started this diet development effort back in 2007 or something, and at that time it was exceedingly difficult to find any existing fish food manufacturer that was willing to develop small batch custom diets for us. I think it's gotten a lot better in the number of years since then, but this was the best that we could do back at that time. So you can see in this panel here, we have a high-fat diet control and a low-fat diet. You can see the amount of dietary fat shown. And of course, we were not able to keep these isocaloric, and so the high-fat diet is also a high-calorie diet. And so again, we looked at gut and water microbiomes using 16S rRNA gene sequencing. And so the impact of these diets on zebrafish growth is shown here at the different ages that we analyzed. And it really wasn't until 70 days that we saw an impact of dietary fat on standard length. And in this case, the high-fat diet that we used impaired somatic growth at 70 days, with low-fat diet having a higher standard length. So in these animals, we looked at their gut microbiomes, and this is how one does that. Generally, you can take a microbial community from any source, you extract genomic DNA from that community, and then using PCR primers that recognize a specific part of the bacterial genome, the 16S rRNA gene, you're able to amplify across genetically variable regions of the 16S gene, and then take those sequences, align them based on similarity, and then define what are called operational taxonomic units, or OTUs, which for our purposes you can think of as being bacterial species or genus. And then based on that information, you can look at the relationship, the compositional relationship or similarity between these microbial communities, either in trees, shown here, or you can take this type of data and convert it into a principal coordinates analysis, which allows you to just graphically look at the similarity of those communities to one another, where the closer two dots are to each other, the more similar those microbial communities are. Okay, so we did that for our study, and here I'm just showing the gut samples, and they're color-coded based on age. So what you can see immediately is that there is a change in the zebrafish gut microbiome over developmental time. So at 5 days, those samples all tend to occupy this space, but then progressively out to later stages, you get a change in the composition of the microbial community. That's what this data tells you. But you can also appreciate that there's a very high degree of inter-individual variation, right? So this is something which is not unique to zebrafish. It's certainly the case in humans. certainly the case in other animal models, but the degree of inter-individual variation is certainly interesting, but a challenge, I think, when considering the interactions between diet and microbiomes. I'm not showing you this color-coded based on diet because at this level of resolution, the impact of high control and low-fat diets did not have a significant impact on the microbial communities, at least with the diets that we used. However, there are other ways to look for more subtle effects of diet or any manipulation on microbiome composition. So what we did was to take all those OTUs, those operational taxonomic units, and we performed hierarchical clustering to determine which OTUs sort of came and went together, right? So which of these OTUs co-occur with each other across the entire data set. And operationally, we're able to define a number of these clusters, which we'll call assemblages. And you can do some interesting things with these assemblages. And I'm just going to show you a couple of examples. This is our analysis of all the assemblages that we pulled out of the study, but I'm going to highlight just a few of you, or a few of these for discussion here. So each row is an assemblage. And the first bit that I want to draw your attention to is here. So this is a blue to yellow ratio of the relative abundance of that assemblage in the gut versus the water, okay? So what you can tell here in this assemblage at the top, which happens to be dominated by bacteria from the genus Aeromonas, this cluster, or this assemblage, was enriched in the gut at all stages, right? So there was proportionally more of this assemblage in the gut compared to the water. So there's something about the gut which somehow enriches for these microbes. In contrast, the one below it, which was dominated by members of the Cingobacteriales, was enriched in the water throughout all studies. Again, so there's clearly distinct microbiome compositions in the gut and the water. This is something that I think has to be taken into account when we're considering, fish and nutrient interactions as well. The second thing that I want to draw your attention to is the last set of columns here, and this is looking at the impact of high versus low fat diet on these assemblages within the gut. And what you can see is that the impact of these diets can have a variable effect on different assemblages at different times. For example, this second cluster of Cingobacteriales was slightly enriched in the gut at early stages, but at later stages was enriched in the water. And perhaps more importantly for what I'm going to tell you next, members of the Firmicutes phylum here, and the last group, cursor's not working, were enriched significantly in the gut at later developmental stages. Okay, and so this raises questions about what are the rules that govern community composition in the gut? How does dietary fat affect those rules, et cetera? These are things which my lab has continued to take an interest in recent years. And so just to summarize this part, so we've seen that the gut microbiome is altered as a function of developmental stage, and also as a function of the individual. There's a significant degree of inter-individual variation, even within animals within a single tank. Dietary fat affects gut microbiome composition as well as the water, and those effects differ at different developmental ages. We can identify co-occurring groups of microbes that seem to be interacting either nutritionally or physically in ways that we do not understand. And those assemblages can vary as a function of age and fat content as well. And the implication here is that dietary nutrients do shape the composition of the gut microbiota and of the surrounding water across the lifespan. Some other brief anecdotes, just I don't have time to go through it, but we've also looked at the impact of starvation, which is obviously a much more severe nutritional manipulation, and it has a more significant effect on gut microbiome composition. And then we've also taken one study in collaboration with Karen Gill and Brendan Bohannon at the University of Oregon to look at how the gut microbiome varies across facilities. And indeed, there are differences between facilities, as one may imagine. Unfortunately, in this study, we didn't control for diet, so but those types of studies have been done. So the next part of the talk that I want to present is looking at how, sort of the other question, how does the microbiome affect nutritional physiology? And we were interested in how the microbiome might affect the assimilation of dietary nutrients. And most of the conversation in this field over the years has been driven by interest in mammals for the most part, and how gut microbes can help liberate calories out of recalcitrant carbohydrates and make those nutrients available to the host in the form of short-chain fatty acids. And this is indeed an important part of host-microbe symbiosis in the gut. But the impact of microbes on dietary fats really had not been well studied at that point. And I'm I'm benefiting today from talking after Steve Farber. You've already heard a lot of this, but dietary lipids dietary fats are broken down in the gut lumen, and through the activity of lipases, their triglycerides are hydrolyzed into fatty acids and monoglycerides, which are absorbed into the cell and reesterified in the ER, as Steve showed us, and those triglycerides can then be packaged into lipid droplets or exported as chylomicrons. And so we wanted to understand how microbes affect this process, and so we collaborated with Steve on this and other projects to use the egg yolk feeding paradigm that he introduced us to, where you incorporate Bethesda-labeled fatty acids into these egg yolk preparations and feed zebrafish. And for this work, we leveraged a set of techniques which my lab has pursued over the years to raise zebrafish in a germ-free manner. So this turns out to be quite easy. We take the traditional zebrafish bleaching protocol, which is in the Zebrafish book, and sort of add some new facets to it, and are able to surface sterilize zebrafish embryos at 0 days post-fertilization, sort of baptize them into notobiotic isolators, shown here, or tissue culture flasks or microfiber plates. And for the majority of our studies in my lab, we provide a sterilized diet, and that diet over the years has primarily been an autoclaved emulsion of ZM000, and then we can colonize with a microbe of choice later on. And most of these studies have been focused on larval stages, including the data I'm going to show you now, but that'll become important at the end. So again, we used these germ-free derivation protocols. We derive zebrafish germ-free at day 0, and then the germ-free animals, we give them a sterile diet and then do this egg yolk feeding assay at day 6. And for control, we can take some germ-free animals and inoculate their water with the microbiome at day 3, and then give them the same sterile diet, do the same experiment at the end. And just to kind of summarize this published data, what we were able to show is that in the presence of microbes, these conventionalized fish here developed more of these Bode-positive lipid droplets within the gut epithelium, suggesting that microbes were promoting formation of lipid droplets. But it didn't stop at the intestine. If we look at fluorescence in the liver, or if we look at total fluorescence in the rest of the body, we can see that there is an increase in the presence of microbes. So microbes are somehow promoting the assimilation of these labeled dietary fatty acids. We were also able in this study to identify at least one microbe that is able to promote this process. And this turns out to be a member of that Firmicutes phylum that I showed you earlier on, that is enriched in the gut by a high-fat diet. And again, we see that Exiguobacterium alone is able to promote lipid droplet accumulation and also transport to the liver in the system. So, from this, then, we're able to pursue, and we are, the mechanisms by which this bacterium promotes fatty acid uptake. I'm not going to go through this whole slide either, but this is just a snapshot of some RNA-seq data comparing the impact of microbial status, germ-free versus CV, or plus or minus these egg yolk liposomes. And I guess really these last two are perhaps most useful. If you compare the impact of liposome feeding, or this egg yolk feeding on colonized animals versus germ-free animals, you can see that there's a lot of shared transcriptional responses, but also a lot of distinct responses. So there are hundreds of genes in the zebrafish genome that only respond to this acute high-fat feeding when microbes are present or absent. So microbes are dramatically modifying the animal's physiologic response to this acute high-fat challenge. This just summarizes that data. Again, we were able to show that microbes promote fat uptake in the system. And the implication here, though, more broadly, is that the presence and membership of the gut microbiome affects nutritional physiology. And so the microbiome is going to be a part of the equation that we're trying to solve here as a group. Finally, in the last minute or two, I just want to highlight a problem that we're facing in the notobiotic field in particular. So the majority of work that's been done in this field to date using germ-free zebrafish has focused on larval stages, and those stages have either not fed anything, or they've fed, as I said, an autoclaved emulsion of a commercial fish diet, which turns out to not be sufficient to promote growth of germ-free fish through metamorphosis. So we really needed some other solution, and we struggled with this for years because all the wonderful biology that occurs in the zebrafish life cycle beyond 5 days of development, we really can't interrogate the role of microbiome on this process without a diet that gets us there. So we've developed one potential solution, and it's certainly not a permanent solution, but this is just a scheme that we've developed here. I probably won't even spend the time to talk all the way through it, but we start feeding at 5 days. We don't colonize until 14 days. I can tell you why if you want to hear more about it. But this is a very laborious process. All the water exchange is done manually because it has to be done in a sterile environment within these tissue culture flasks. And we're very careful to monitor density and media following. The diets that we use for this are both live sterile diets. So we've tried some autoclaved and irradiated chows from different providers. Those really didn't seem to work for us. And so what we do is we raise germ-free cultures of Artemia and germ-free cultures of Tetrahymena and feed those starfish. And the growth is slow, but you can get animals that make it through metamorphosis into adult stages as germ-free animals. However, there are problems with this. So survival, shown here in the bottom left, is relatively poor through the metamorphic stages, with colonized animals having lower survival rates than germ-free. And furthermore, another challenge we have, although there's obvious biological interest in this, is that germ-free animals under this protocol, when they get to about 30 days, uniformly, perhaps in a standard length-dependent manner, they develop brain edema and hemorrhage, which eventually kills them by 40 days of age. We don't know what the nutritional basis of this is, but it's something that one could interrogate if we had better diets, I think. So again, as we think about how to understand the nutritional requirements of conventionally reared zebrafish, I would argue that, at least for my part of this field, there's a lot of interest in understanding how those nutritional requirements change when microbes are not around. These are just the take-home messages. Again, I won't talk all the way through it. I want to acknowledge the people that did the work, though, even as Simona, Sandy Long, and so Gomez, Delatory, Candy were primarily responsible for generating much of the data that you saw today, and I'll leave these up for discussion. Thanks. I think I have like 1 minute for questions.
I guess I've always assumed, but is it true that a tank of fish next to another tank of fish is going to have the same microbiome, or like, can you?
Thank you. No.
Sorry.
Don't make that note. So that was one thing that I skipped over here. Yeah. So in all of our experiments, this is important, especially for those of you that review papers. We see a non-trivial amount of inter-tank variability, even when the animals are given from the same clutch, same facility, presumably the same diet, it's right next to each other. There are some stochastic processes that can occur during the assembly of tank microbiomes and fish microbiomes that are non-trivial. So it is improper to do a study where you have one tank with condition A, one tank with condition B, compare those and assume that the microbiome is equivalent across. You really have to do experiments where you have multiple tank replicates per condition. It's really important. Thanks. Steve?
In the mouse community, has anyone figured out maybe these magic micronutrients or something that bacteria make for the host? And,
right.
And those were what's missing in the fish? Do anyone know like the class of molecules, you think, or is it really an unknown world?
There are some good candidates. I should probably know more about this than I do, but there are a number of vitamins which are required, which in the absence of microbes may be deficient. We've tried supplementing vitamin mixes into our diets, and those didn't seem to really do the trick. But there is a rich literature. It's very dated literature, but it's a rich literature on this. One of the interesting things, and then I'll close, is that, I mean, as you look at the microbiome field in general, if you pick up the last 3 issues of Science or Cell, what you'll find are some microbiome papers that have discovered a new chemical, a new nutrient that microbes produce that are important for some aspect of the host phenotype. And really, even studies in germ-free mice have been using some of the same axenic diets for decades, right? And so one of the ways that you can view the field is this gradual, continued realization of new, unappreciated chemical and nutritional contributions of microbes to normal host physiology, which, despite those discoveries, those nutrients are not necessarily being incorporated into new versions of the germ-free mouse chow. So that's a struggle that hopefully the field will continue to pursue because it's important science. But yeah, and I'll stop there.